This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 693, recorded on December 11, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. It's 50 Fahrenheit, 10 Celsius, clouding over, supposed to rain tomorrow. And fortunately, the stump grinder that was running in the neighbor's yard is now all done with the job. But I just wanted to mention that because longtime listeners will appreciate the stump grinder arc.
So you didn't want to have a stump grinder as our background music.
That would have been some distracting background noise, yes.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is 54 Fahrenheit, 12 Celsius, and sunny here. Big change from Wednesday when we talked.
Yeah, it snowed a little, right?
It did.
It's all gone. And from Austin, Texas, Rich Condit.
Hi, everybody. We've got, what is it here? Sorry. 67. Overcast. Drizzly. And to Brianne, a lot different than it was when we talked.
A lot different than it was. And from Fort Lee, New Jersey, Dixon Despommier.
Hello, everybody. It's the same here as it was when Brianne spoke. Whatever she said.
The Aaron Diamond AIDS Research Center at Columbia University Irving Medical Center, that's where I am, is seeking a high biocontainment laboratory assistant manager to help run the day-to-day operations of the center's biosafety level 3 core facility and to assist investigators in SARS-CoV-2 experiments. Applicants should have practical experience in performing cell culture microbiological techniques and basic molecular biology. Applicants must have a bachelor's degree in science with 3 years experience working in a BSL-3 or 3 years of non-BSL-3 experience and a willingness to travel for BSL-3 training. And I'll put a link in the show notes. You can apply to that. We've been very good at recruiting people for various positions. The other Institutional Biosafety Committee position, not committee, but you know what I mean.
Yeah.
The Institutional Environmental Health and Sciences, let's put it that way.
The biosafety job, right.
Biosafety job. And also Amy, a technician for Amy. We recruited both TWiV listeners.
Oh, you got one. Good. Great.
And Amy's tech has come from Seattle. Welcome.
My goodness.
She's a
Great.
TWiV listener, so maybe she's listening, or probably Amy's keeping her too busy now. So there'll be a link for the show notes for this BSL-3 position. And I think, Dixon, you could apply for that. You've got cell culture experience, right?
I do. I've cultured all my cells, in fact.
A little bit of news. We have two papers quite interesting for you today. A little bit of news first. As everyone must know, the FDA Advisory Committee yesterday voted in favor, 17-4, right, for the FDA to make an EUA for the Pfizer vaccine?
17-4-1. One abstained.
One abstained? Like what's the abstention about? I don't get that.
I think
maybe they felt they had a conflict of interest?
I think all of the no's as well as the abstention were about the 16 to 17-year-olds.
Yeah.
As opposed to it starting at 18.
So abstention is a way of saying, I don't want to say no, but I object to this part of it, maybe, right? Anyway, it's good enough for the FDA to take note. And in fact, they're saying they're going to approve it tonight by the end of Friday. And so next week, the vaccine might be rolling out, which is cool. You know, it's the first phase here in the U.S. of getting something going.
Here.
I'm very excited. I think it's good stuff. And
I'm not sure where I stand on the 16 to 18-year-olds because the issue is they weren't in the trials, but the reasoning is, well, like close enough.
There were a few people in trials, very few, apparently.
Yeah.
And not enough.
Yeah,
that was the objection. The 4 people said there are not enough kids to really make a decision.
Yeah, and they're, I know Moderna is starting right now. They are starting a trial on, I think, 12 to 17-year-olds. So the expectation is that this is going to get tested, and that obviously, since it's already been tested and proven effective in adults, you're not as concerned. And then you go into kids. That's why you don't start in kids, because it's a worse place to have a problem. But I think the expectation is that, sometime in the spring we'll get an answer on efficacy in kids.
Yeah, so this is only an emergency use anyway,
Yes,
right? This
absolutely.
Yes.
So there's another date for the actual approval for everybody. So
Yes.
does anybody know whether in England they raised the same issue before they approved it?
The issue of what? The young age?
So, yeah, 16 to 18-year-olds, yeah.
I don't know if
'Cause
they approved it for that group.
I don't know.
Well, that's right. I don't know any details of that. I thought,
Yeah.
but
Yeah,
no
I'm not
issues
really
were
sure.
raised.
They also gave an emergency approval or the equivalent of it.
Yeah, that's
And
right.
I don't know the details of it.
The Pfizer said it will apply for full approval in April after they've collected 6 months of safety data, and then they could sell it to anyone who wants it. Right. But right now, emergency use. And so as we've talked about, healthcare workers, elderly people, et cetera, first. And by the way, speaking of getting in line, the Times has a vaccine timeline. You can go to it and put your age, your occupation, comorbidities, and it will tell you how many people are ahead of you in line. And I am 180 million behind.
I think Brienne's probably going to win this, but I've got 268.7 million in front of me.
Yeah, I'm putting it in right now.
It's pretty funny.
My wife, it says, it doesn't even get a number. It just says very few.
She's a healthcare worker. Yeah.
Worker.
Very good. So she should be able to get it in the next couple of weeks.
She should be
right?
the first one in the household vaccinated. My daughter is 15, so she won't get it till they approve it for kids.
Yeah. That's very
Or
cool.
until she turns 16.
Dixon, you ought to put your numbers in, Dixon, because...
Oh, look how far back I am in this line.
How many?
You're behind me.
268.7 million.
Oh, we're in the same group. I see. Okay.
Did
Guys
you
are
look at the
young.
picture at the bottom?
Yes, it goes
There's
on.
actually a line that shows
Yes.
people and where you are in that line. And I have to scroll quite a while.
Yes.
It's very funny. Dixon must be higher up because you're so ancient,
He must
Dixon.
be. I don't want to reveal yet what's happening here, but okay. I just
And Condit is going to be more or less like me.
I'm 118.5 million in
Oh,
line.
he's ahead of me. Cool. And maybe it
I'm
goes by
older.
state. Do you put your state? Oh, you're older.
You
That's
put your county.
Put
Yeah.
your county.
Put your county.
I'm only behind 7 million others in New Jersey.
Oh, that's not bad, actually. It only
I'm
takes
only
you
behind
6 months.
5.4 million in Massachusetts.
This is very fun. It's a lot of fun. Amusing. I'm just going to stay in here and podcast for the next year, and I won't worry. But what is this setback of the Sanofi Glaxo? Does someone have?
Oh yeah, so I popped that in there. Let me get back to the notes here. So Sanofi Glaxo, which I'm really disappointed about this because I was looking forward to this one. It's a subunit vaccine. Uses the same technology as one of Sanofi's flu vaccines. And if you didn't know, Sanofi is the world's leading supplier of flu vaccines. So if you got the flu shot, as you should every year, you have probably gotten a Sanofi shot. I know I did this year. They and Glaxo have been collaborating on developing this. They were planning to start a phase 3 trial this month. And being Sanofi, since they operate at such a massive scale, they were expecting to produce 100 million doses of vaccine by the end of this year. So this month. And they were going to make 1 billion doses in 2021. This is a vaccine that can be stored at refrigerator temperatures.
Wow.
So this is kind of, vaccinate the world type stuff, both in scale and in storage. The problem is that they had, stop me if this sounds familiar, they had a dosing miscalculation in their phase 1/2 trial. And of course, this also happened with another trial.
Yep.
So some people ended up getting a lower dose than they should have because there was an error in measuring the dose that they were supposed to get. The adults, ages 18 to 49, demonstrated a fine immune response based on that, but the folks over 60 had a weaker immune response, weaker than people who'd recovered from COVID-19. So then they're going to back up. I think they're doing a phase 2B or something just to back up and redo that, make sure everything's good. They're expecting to start their phase 3 then next year. And now they're looking at late 2021. The interesting twist here for approval, if everything goes well. The interesting twist here, though, is they're saying now, since this has slid so much, obviously we're going to have Pfizer, Moderna, possibly AstraZeneca, maybe somebody else on the market by then. So then who's going to want this? Right. I mean, we're piling up the vaccines. Well, but who's going to want the fifth one on the market that, comes out in November of next year after everybody's already gotten their shots from other companies? And while there will, I think, still be a sizable portion of the world's population that'll need it, the interesting angle that Sanofi is taking on this is, well, how about if we do a comparative trial? So they are deliberately setting themselves a higher bar and saying, instead of doing a placebo control, because the other issue is, how are you going to start a phase 3 trial in an environment where people have access to a working vaccine?
Yes. Yeah, you can't get rid of standard of care, right?
I mean, I'm very pro-science and I want to see these trials work, but there's no way I would volunteer for a trial that might give me a placebo when I can go and get the real shot against this virus.
It's called standard of care, right?
Yeah, so standard of care, the idea is to take people and randomize them into either one of the approved vaccines or Sanofi's vaccine.
Yeah.
And then see how that works out. And they would have to either match or beat those vaccines, which would be cool, And so I appreciate them doing that, and I hope this ends up working out.
So I understand they were the sixth company engaged by Operation Warp Speed, right?
There's sixth. I lose track.
Yeah, I think so. Okay.
So, Vincent, my number is 283K.
Wow, pretty good.
283,000. Okay.
Yep. So I can see the end of that line. Wow. I can see the end of the line from my house.
What is it in New Jersey? I bet you're pretty high up in New Jersey, because that's U.S. 280.
That was New Jersey.
Ah.
All right, so pretty soon you'll be able to come in here, Dixon. Let me ask you a question, Dixon.
Yeah, please do.
Oops, I got Rich there instead of Dixon.
That's okay.
There's Dixon. All the labels are wrong. Sorry, folks. Dixon.
No
Here's a question. Once you get your vaccine, can you throw your masks away?
Absolutely not. Why would I want to do that? I can still. Let's see, what can I do with my mask? I can show people that I still care whether they get infected or not.
I'm just using this opportunity to look at your fish. That's all. Sorry.
Oh, all right.
Okay.
I can trade more pictures if you want.
Yeah, another time. We saw them at the end once of some.
We did.
Thing. Actually, speaking of vaccines, one of our papers is about vaccines, and in fact, we were talking about the AstraZeneca ChAdOx1 vaccine last Friday, and people asked about data, and they said, well, it hasn't been published yet. And then after TWiV on Friday, it was published. And
right.
Kathy pointed it out on Wednesday, so I thought we'd take a look at it. It's published in Lancet, Safety and Efficacy of ChAdOx COVID-19 Vaccine, AZD1222 against SARS-CoV-2, an interim analysis of 4 randomized controlled trials in Brazil, South Africa, and the UK. And of course, we know the results already. This is the one where some people got a half dose first time and so forth. We know the numbers, but I want to go over a few things that are quite interesting and you might not have seen before. And of course, this is a very big author list collaboration between University of Oxford and AstraZeneca and many other places who've participated in the clinical trial. And the reason I want to do this is in particular to take a look at the side effects, okay? Because they have an appendix document here that has all the side effects. And we'll get to that. So this was done in 3 countries, right? UK, Brazil, and South Africa. And they pooled all the data to get at their final efficacy numbers. And of course, they had this one group where they got, everybody got 2 doses, but one particular group, the first dose, which was made by a contract manufacturing organization, right? By somebody else, in other words. They used spectrophotometry to quantify the amount of virus, and it turned out to be different from PCR quantification. So they ended up packaging half the dose. That's what happened, right? And
and this reminds me of trying to get an accurate read on my solutions of RNA and DNA, okay, which absorbs at 260. And you got to be careful that you don't have anything in there other than RNA and DNA that might absorb at the same wavelength. In particular, a reagent that's used commonly in our procedures, beta-mercaptoethanol, that absorbs very strongly, okay, and can give you a false high reading. So you think you've got a lot when you don't have as much. And they talked about in this particular process, they didn't discover until later on in the quality control that there was an excipient. I had to look it up. Okay. That is something, some other reagent, probably innocuous. Okay. But that absorbed at whatever wavelength they were doing their photometry and gave them an artificially high reading. Hey, got to be careful.
And well, this is what happened with, as far as I can tell, with the Sanofi situation. It may even be the same thing for all I know. And it raises the important point that this is a new virus, right? And we are accelerating it's being done as safely as possible, but some of the steps in the protocol, little things like this can suddenly say, oh, wait, we didn't check that, did we? So,
well, if you spent I've kind of shadowed my ex-postdoc, Susan DaCosta, as she moves through biotech, okay, and learns what this is all about, and encounters the rigor of standard operating procedures and quality control and all the rest of it. And it's a discipline and a skill all by itself. And this just goes to show you how important that kind of rigor is.
Yeah. And they caught it.
Yeah, they caught it.
I mean, they got partway into this trial, and they had given some people initial doses, and it's not a large number of people who got this low dose. It was for a short period of time that they were dosing while they were checking, triple checking everything, and said, "Oh, wait a second," and then, fixed it. But as a result, they've got this subgroup that got low first dose, higher second dose, and it actually turns out to be kind of interesting, as people have probably already heard.
And if I remember correctly, the way they caught it was that the people who were getting a lower dose did not have as exaggerated an initial reaction to the shot, and some alert physicians said, or people running the trial said, that's funny. These people aren't reacting the same way as the other people. I wonder what's going on.
And so the people who got this were in the UK and in South Africa, not in Brazil, as we originally said.
Right. And I also thought there was an interesting part in the description of the protocol where they talk a little bit about the fact that sometimes they were not able to do a perfect 4-week later boost. And so they also spent a little bit of time thinking about, how long after the first dose was the second dose given. And I think that that's going to be important sort of practically as we move forward, because that may be what happens with some people in real life.
And in fact, some of the issues they had with the second dose are things that we may very well see, because they had a supply issue, I gather, at one point, and it delayed people's doses by a couple of months. So there ends up being a much bigger gap than was originally planned for some of these patients.
They even wound up in... Go ahead, Dixon.
Go ahead. Well, I was just going to say that Pfizer has already said that it doesn't really matter much if you get your second shot second week or third week. So there's a lot of wiggle room in the second dose, apparently.
There was enough concern about this, or interest, shall we say, in this, that in the paper they wound up splitting up the different boost times or intervals, okay, and comparing the results in each of those groups. And one of the things I found amusing was that they had a way of saying in the paper, in this case and in others, that we're presenting this because the reviewers asked us to.
Yes.
Yes.
Nice. So the, as I said, UK and South Africa, some people in those arms got the half dose. Then a couple of other things I found very interesting. So first of all, they immunized people with vaccine or with placebo. Then what did they do? Participants were asked to contact the study site if they experienced specific symptoms associated with COVID-19. And they got regular reminders to do so, because you can imagine you might not remember. Those who met symptomatic criteria had a clinical assessment, a swab taken for some kind of nucleic acid test, and then blood samples, right? So if you have cough, shortness of breath, anosmia, or ageusia, you would have, which is loss of smell and taste, right? You would take a swab, and then they would see if you're SARS-CoV-2 positive, because they want to confirm it by after the symptoms, right? So that's how this works. And then they say, we're counting the people with confirmed COVID-19 in the vaccine and the placebo. Although at the time that the data are collected, of course, you don't know who's who. You just know there's persons in the trial. And then later, when it's looked at by the Data Safety and Monitoring Board, they get to see who's who.
Yeah.
And they also did screening for asymptomatic infections.
Yes.
Which I appreciated a great deal.
So they were asked to provide weekly self-administered nose and throat swabs from week 1 after the first vaccination. They were provided kits for this. And this is very interesting. We're going to look at the results from that, which are quite interesting. So this was done, it was not done in Brazil, they say. So
right, it wasn't everybody who had that.
And let me see, what were the 4 sites again? UK, Brazil, South Africa. Just 3 sites? Excuse me? 2 in the UK.
2 were in the UK.
Because there were 4 trials, right? 2 of them were phase 1-2. 2 of them were phase 3. And they pooled all 4 for the safety data, which is perfectly legit. And for the efficacy data, just the 2 phase 3 trials where they assessed efficacy. So there's different numbers in those different assessments.
The other thing they did with the efficacy trial to part of the trial to keep the data clean, you want to cast as broad a net as possible to find any problems, which is why you look at the safety data on pooled. And then the other subdivision they made in the efficacy is if in the analysis, if they were seropositive for SARS-CoV-2 at baseline before getting the shot, so they'd been infected before, they could still go in the trial and they could still get the vaccine, because you want that. But they were excluded from the efficacy results. So if you were in the trial and you got the vaccine and you'd previously showed antibodies to SARS-CoV-2, then your efficacy to it, that's nice for you, but it doesn't count for the trial, which is an important point, because that could really skew your results otherwise.
Yep. And the other thing that they do that we should just point out here, so I listened to the FDA meeting yesterday, and one of the things that came up in some of the discussions were that the placebo group was getting saline. And so they assumed that people were able to identify whether they were in the placebo group or the vaccine group, and that perhaps that changed their behavior. In this trial, instead of using sort of saline as their placebo, they used another vaccine. They use a meningitis vaccine. And so if you were listening to that yesterday and heard about the concerns about people self-identifying as placebo based on reaction to the vaccine, that would not be an issue here. Both groups could have had a reaction to their shots. It was just a question of whether they're SARS-CoV-2 or meningitis.
And there's another reason why that's important, and I kind of prefer this trial design. There's evidence that's been kicked around and that we've even talked about on TWiV that maybe just kind of generically goosing the immune system with something like BCG or polio vaccine or whatever provides some level of protection. And so you'd hate to put all this money and effort into developing a very specific SARS-CoV-2 vaccine and find out that everybody could have just gone and gotten their flu shots and it would have worked, right? So it's nice that they've got a real vaccine as the control in this trial. And who knows, that might even explain why they see 70% efficacy versus the other trial seeing 95. We may never know if that was.
Yeah. Well, what you can say is that this meningitis vaccine doesn't protect you, because the placebo had most of the COVID cases.
It did, but maybe they would have had even more if the placebo was saline.
Could be.
Is the point.
Right.
Could be. Well, we could compare it to the mRNA vaccines where they have used just saline, right?
Exactly.
Numbers are similar, right? 90%. 23,848 participants all told. And a couple of things that I wanted to point out. Well, first of all, the standard two dose, we've given these numbers before, 62.1% efficacy. And then the high dose regimen, 90% efficacy. Here, a small portion of the participants were seropositive at baseline. And that's interesting. People have often asked us, if you're seropositive or been infected, are you excluded? And apparently not. They just enrolled everyone because I think they don't do the serology until you're after enrolled and already got the shots. So this is looked at in retrospect, and I'm not sure it made any difference, right?
Well, and you also want those people in the trial because a lot of folks have had it, don't know it, will go get the shot. And you'd really like to know if you have some issue or, is it a good or a bad thing to get the shot if you've already had the virus? And in this case, it seems like probably fine to go get the shot.
138 people in the UK and 235 in Brazil were seropositive, at baseline. Yeah. I don't know what happened to them. I'm sure we could find it in the data. But
3 participants seropositive at baseline had a subsequent PCR positive swab.
So one that means they got infected.
They got infected. One got 3 weeks after the first dose. The other two other participants in the control group had symptomatic infections 8 weeks and 21 weeks after the baseline sample was taken.
So they weren't protected, basically, right? They were not protected.
It's another example of the issue that natural infection does not seem to be a reliable way to have immunity against this virus.
It's quite interesting. It's only a few out of the hundreds.
It was only a few, yeah.
I
mean, and across the board, we're talking about small case numbers, and I think the big one, this low dose, standard dose, the people who got the low dose first. There's a lot being said about, well, it was 90% effective in that group. Well, yeah, but you're talking about 3 people in the vaccine versus 30 people in the control group got the virus, and the confidence interval on that is really wide, and this is a tiny number of cases, and so there's some concern that maybe even with that, it's not quite right.
So for all the people who in some corners of the internet are really worried about vaccines potentially leading to enhanced disease if you've already been infected, this is certainly not, all of the data you would want on that, but this is some data to indicate that the individuals in this trial who were in that situation were okay.
Yeah. And I think we have similar findings from the Moderna and Pfizer trials, don't we? Or did they exclude seropositive people?
I don't know. Good question. I would have to go back because those have been published, right?
Yeah, I think they included them, but...
Actually, they haven't been published. I'm sorry, right? The Moderna
they have not been published.
Phase 3 have not, yeah.
But, one of a major concern with the enhanced disease issue is that if you get vaccinated and then get a natural infection, might you get worse disease?
Right.
And I think that if that were happening at any largish frequency, we would know it by now.
Yeah, given the size of these trials and all 3 of these trials taken together, that's just not showing up.
It could be 1 in a million, though, right?
It could be 1 in a million, and that's why the full approval for Pfizer, is waiting for 6 months of safety data because they want to roll it out for emergency use and see how things go and monitor it all along.
So let's talk about these asymptomatic infections, right? They were asymptomatic or unreported systems in 69 participants. Okay, so then they could calculate the vaccine efficacy with the idea that this is a small number, right? In the 24 low-dose, standard dose recipients was 58.9%. In other words, how good was it at preventing an asymptomatic infection? Whereas it was 3.8% in the 45 participants who got standard dose. Both not great, right?
Right. So it seems like this is a vaccine that's preventing disease more than preventing infection, but it can prevent infection in some group of people.
And if you get the 2 standard doses.
This is why after Dixon gets vaccinated, he's going to have to continue wearing his mask.
Exactly. Yes, because
they did find in this group, in the folks who got the virus, tested positive after they'd been vaccinated, they didn't get very sick. In fact, I think none of them were hospitalized, isn't it? Versus a number of hospitalizations in the placebo group.
Anyway, that's interesting.
So it does seem to be very protective against what you're really afraid of, which is getting very sick or dying.
Good enough for me.
But it needs, you can still probably transmit, and so
you can still probably carry it.
Yeah, that's right.
Until a good number, a fraction, 70 to 80%. Peter Hotez said last night he said we did those numbers here in Houston. 70 to 80% of the population is immune. Then you might be able to. Then that would inhibit transmission sufficiently because
that would have to be sterilizing immunity.
No, it doesn't have to be. The number of people that now protected. Oh, I see what you mean, because they're still going to be infected.
Yeah.
It
doesn't matter. You could vaccinate if you had a vaccine that was purely protective.
That's true.
And not sterilizing. You could vaccinate 100% of the population. And then one person comes along who's not vaccinated, they could get the virus and die.
Yeah, because those 100% are still shedding, you're saying, right?
Those 100% could still be spreading it like crazy.
Well,
and probably would be if they'd be safe.
According to the numbers here, if you do the low dose, high dose, what is it, 58%? So that leaves quite a large percentage that is not protected and still going to be shedding.
You can't get to herd immunity with this if those numbers hold in a larger population.
Yeah, that's right. It's very interesting. So what's the outcome of this? We have to wear masks forever?
No, the outcome of this is you're going to have to vaccinate everybody. And if people are not vaccinated, they're going to run the risk of dying of this virus.
That's right.
And everyone means as far over 90% as you can get, right?
Yeah, right. It means everyone. It means, like, how many people do we have in the world now? 7 billion? Pushing 8 billion?
7.8 billion.
Closer to 7.8.
It's a lot of doses. Wow.
Yeah. It's a lot of doses.
Yeah. It also means we might need some of the things like distancing and things like that as longer term changes.
Yes.
Okay. The other thing I wanted to discuss well, first of all, there's an appendix, 2 appendices, and one of them, table S4, are the baseline characteristics: age, sex, BMI, ethnicity, percent health and social care workers, comorbidities I wanted to point out. So the 3 comorbidities that are represented: cardiovascular disease, respiratory disease, and diabetes, right? So those are people at risk for serious disease, and they want to see if the vaccine is okay in them, and it is, right? That's important. So that's only those 3, but that's important. The other is the side effects, because a lot of people are concerned about side effects. And what they do is they have a huge table where they have serious adverse events, because, the moderate ones: fever, pain at the injection site, fatigue. We talked about this on Wednesday, a third of the control people are fatigued because people are just tired. And all those things are not game-changing side effects, but the serious ones, right? Those are the ones. And there's a big table because, as they said, some people died during this trial from a traffic accident, blunt force trauma, someone hit them over the head, or homicide, or fungal pneumonia, right?
Probably not caused by the vaccine.
Not caused by the vaccine, but when you immunize, 30,000 people, or you put them in a trial, things are going to happen to them. It's
right, and you have to report all of that.
Yeah. And this table, S6, serious adverse events by organ class, very granular, and it goes on for 6 pages. But what they do is divide them into the events in the vaccine group and the events in the control group. So you go down and oh my gosh, 5 cardiac disorders. Yeah, there were 6 in the control group too. And you go down all those gastrointestinal disorders, same thing in both groups. Injury, poisoning, procedural complications like animal bites. There was one in the control group. I guess the vaccine prevents against animal bites.
Yeah.
Bone fractures, other infections. What else? Metabolism disorders, cancers. 4 in the vaccine group and 5 in the control group. The point is, when you hear about side effects, in the vaccine group, you have to look at these charts in the control group because people tend to leave that out. I heard the other day someone said, oh, there was Bell's palsy in this trial and 3 people in the vaccine group. Yeah, but there were 3 in the control group too.
Right.
So that's the key. And most people, I think, are not going to go through this table and look at it. But in every case, the serious events happen in both the vaccine and that's why, and the control group. And that's why they say they're not related to vaccination. Homicide, yeah. There was 1 homicide in the control group. So all the things that happen to people, here they are, and they're in both groups. So that's why they're saying so far after 2 months, this thing has a good safety profile, right?
Yeah. And by the time most of us who are 200 million people behind in the line get this vaccine, we'll have a lot more safety data and a much bigger.
That's right. Yes.
But even if you're at the front of the line, I think there's good reason with this and with the other 2 vaccines. I feel pretty confident in them, at least for protecting against disease.
Now, go ahead, Rich.
I just wanted to point out something that I've been thinking about in these data. And get your reactions to it. My bottom line is that I want to see more data.
Sure.
Because, for example,
before what you get immunized, or just in general?
I want to see more people sick. Okay, I'll give you an example. In the low dose, standard dose thing, where we're saying the efficacy is 90%,
right.
The total number of people who experienced disease was 33.
Yes.
In the trials that we've been evaluating so far. They've been saying that their cutoff for being able to get a statistically significant number is somewhere over 100.
Yeah.
150, sometimes on the order of 200. And here we're talking about 33 cases. So based on the standard that's been set for the other vaccines that I've seen, and I'm no statistician, but that is lower than what would have been an acceptable number. So in order to, it's been widely touted that this low dose standard dose protocol gives 90% efficacy. I want to see more cases.
Yeah, right? That's why I called that out. That's why I called that out earlier. And they're pretty forthright about that in the paper.
Yeah, absolutely. Nobody's trying to hide anything.
Right. No, this is a small number. The one thing they didn't mention in the paper was that, the details of how this has been hyped. But I think the researchers themselves are very, aware that this is a small finding so far and that this needs to be extended in a whole new trial, most likely.
Actually, if you subtract those 33 out of the total number of cases, you've got less than 100.
Yes.
To get you to the 70% number, and that's really borderline. So we need more data on this.
Sure.
We also need to know how long this lasts, right? Because it's only 2 months, and we need to follow people for at least a year to know, are we suddenly seeing infections, or do we need a booster? That's really important to do. So
and all these in the show where I summarized the meeting about all the trials, these are all being set up with 2-year follow-up planned. So that will presumably get at that question and also longer-term issues in general.
Yeah, and I want to, agree with Alan here that in the paper they're totally transparent about all of this stuff.
Yeah, of course.
Okay, it's all there for you to see.
There's one more thing I wanted to bring up that's really, all right, duration, that was one thing. Oh, here it is. I want your opinion. So this trial or some other trial is 90% efficacy. The FDA issues an EUA. What about the control? Why are they not getting the vaccine? So do we break the trial or do we go on? The FDA hasn't decided what to do. What do you think?
That was also discussed at length yesterday.
Yeah. Mm-hmm.
Yes.
What did they decide?
And they were discussing basically that the controls would be offered vaccine sort of when their priority group was getting vaccinated, but they were going to not break the randomization. So they would call in everybody in the trial when their group came in. And if they had previously been a vaccine recipient, they would get placebo. And if they had previously been a placebo person, they would get vaccine. So they were still blinded to themselves, and they could do an analysis of sort of what happened after vaccination versus when you were still a control person. And there was even a fancy name for that kind of trial that I don't remember right now. But that was actually one part of the discussion that did go on for quite a while.
Great. Very good.
Brian, good for you watching that whole thing. How
Great
long
show.
did it go on?
I didn't watch the whole thing. It went from 9 to 6. And I did not make it all the way through.
So I didn't
I
watch
watched
it.
a fair amount of it.
I didn't
I got
watch
the
it,
notification
but,
about it. I was like, I'm not making it to this, but thank you.
It was a very good thing to turn on while you were doing, other work in your emails and other things. Yeah.
At 5:30, I was scheduled to do a TWiV with Daniel, so he's not showing up. I was like, "Hey, you come?" And he goes, "Just a few more minutes. They're voting. Hold on." I said, "Yeah, I got it. Come on." Then he came on. And he said, "All right." I said, "What's the result?" He said, "I'll tell you during the recording." He wouldn't tell me.
Oh,
All right. We have another interesting paper which has a couple of cool aspects to it that I think you will like. It's published in Nature Microbiology. Therapeutically administered ribonucleoside analog MK4482/EIDD2801 blocks SARS-CoV-2 transmission in ferrets. And 3 authors. Holy cow.
3 authors on a SARS-CoV-2 paper. Is this legal?
Is it legal? Right. Robert Cox, Joseph Wolf, and Richard Plemper, who was on TWiV earlier this year. I visited Georgia State last year, at the end of last year. He was on a TWiV with some other individuals, talked about antivirals. And so this is from his group. Very interesting. So the story is here. This drug, MK4482, we'll call it that, is a prodrug of a nucleoside analog. And I'm going to show you what this looks like because we have tech here. Where is this? Here we go. Look at this tech.
Nice.
I could move it down so I don't cut anybody off. Yeah, let's cut me and Alan off, right? How's that?
There we go.
Everybody looks better. So here's MK4482. It's a prodrug. What does that mean? Well, these are orally delivered drugs. They're taken up and then they get modified to form the actual drug. So this is a prodrug of N4-hydroxycytidine. Okay, let's go all the way to the right. I'm sorry for people who are just listening. I'm showing a picture of cytidine, one of the 4 bases that goes into RNA and DNA. There are no phosphates on it. It's just a ribose and the base, right? And there's the site. What is it? The base alone is cytosine. Is that right?
yes.
And with the sugar is cytidine. Is that correct? And it would be
I believe so.
cytidine.
You just stumped everybody with a biochemistry
Let's vote.
That is correct.
I think this is an important thing to know. And then cytidine triphosphate, right?
I know I
It is.
had to know it for a test at some point.
This would be one of those moments where I would say, well, students, I was waiting to make sure that you knew the answer.
Yeah, that's
why-
right. That's right. While you look it up. So cytidine has the ribose sugar, 2 hydroxyls. It's here. And then the base is up here. And you notice this has an amino group at the top, right? So the actual drug, N4-hydroxycytidine, it's exactly the same, except it's got a hydroxyl on the nitrogen. And that's it. And that's enough for it to be inhibitory in a mechanism we'll say in a moment. The prodrug has a little extra stuff on the ribose. It still has the NHOH on the cytidine, the cytosine, but it's got some extra, what would Rich call it, spinach or something on the chicken wire? Spinach.
Yeah.
So this is, chicken wire here, as Dixon would say. It's got some extra, and that's cleaved off when it gets into you. I don't know if it's cleaved off in the gut or in cells. It could vary.
My money's on the liver.
Liver?
Yeah, that's where most.
Yeah,
of
the
it
liver is the master chemist for all of this kind of stuff.
And this is a very, very common theme in pharmacology where the drug that you take is not the active molecule. Your body converts it into the active molecule.
The drug you take is not equal to the drug you make. Is that a song?
That's exactly right.
Beatles. That was the end of the Beatles.
So you take it up, it goes into, you take it orally, which is nice here. This is an oral drug. And it's not a drug yet, it's a candidate. But then it gets in the circulation, goes to the liver, it's modified. Then this gets into cells, and in the cell it has to be triphosphorylated by cellular enzymes. Otherwise, it cannot be incorporated into RNA. And in general, molecules with phosphates don't get into cells very well unless you do funny things to them. And So this is an example of that. Okay, so that's
and
the
this
drug.
This drug was previously developed as an influenza inhibitor. This isn't some brand new thing that they came up with for SARS-CoV-2. But the oral bioavailability is really a very important point, because the only thing, the things that we've found that have efficacy against SARS-CoV-2 so far are all IV, or have other
exactly right. That's exactly
have other
right.
issues, which is fine if you're, a highly placed government official who gets gold standard health care and rushed to the hospital as soon as you test positive and you get injected with all the latest stuff. But if you're some ordinary schmuck like us and you don't show up to the hospital till you're coughing your lung up, then an oral drug would be better.
Or you could be a celebrity lawyer, right?
Any of those, yes. If you're in the news enough, you get higher standard of care. But this is something that, if approved as a drug, which is still a big if, as we'll probably discuss, could be handed out much more easily.
Right. And that becomes really important with the timing issues that we've talked about before.
Yes.
We've talked about how some of the other drugs are maybe less efficacious than they could be, because they're only being given to very severe patients who are already in the hospital and who are already hospitalized. And if they're given IV, you kind of need to be in a hospital for them.
Right.
This is something that could be given to people perhaps earlier at home to many more patients.
Dixon, you were going to say something?
I just wanted to say what happened to the hydroxychloroquine ARC that started so robustly as a central administration recommendation for being treatment, and virtually nobody in that administration ever took hydroxychloroquine as a curative, and they were so adamant about how effective it might be. I'm just
it's dead in the
It
water.
didn't pan out.
It was trialed and it doesn't work. It doesn't work. In fact, it harmed some people.
Yeah,
but there's still people that are touting it, though.
Yeah, but they're wrong. They should move on. Let's move on. You know what that phrase means? Move on.
Move on.
No idea.
So the IV business, even for the monoclonals, it's hard because they have to be given IV. But now they're making infusion centers outside of hospitals to try and deal with that. You can go in for 2 hours and get an infusion, and you have to be in a certain risk group. So you have a positive test and you have a certain age, comorbidity, then they'll send you to get monoclonals, and they're hoping that helps because it's a little earlier than being in the hospital, right?
That'll probably help a few people. And again, if you're in a position where you can just rush out and get this kind of treatment, then lucky for you. But but a pill is what you want, because then you can call up your primary care doctor, say, "Hey, I just tested positive for COVID-19." They say, "Okay, I'm writing you a script. Go to CVS, pick it up."
Yeah, it's like Tamiflu right now, right? Or Lenza.
Yeah, exactly.
You get a script, you take it. So this drug, as you'll see, is really good for ferrets. And as we know, ferrets are not humans, so that has to be tested in people. But ferrets, and here's one of the things I wanted to point out. So they infect ferrets intranasally. And ferret is a mustelid, which includes mink. So we know a weasel genus. And here we go with the stoat again.
Okay.
Didn't we go through the stoats before?
Are we going to start a stoat arc?
I already forgot what a stoat is. It's fine. But anyway, they put the virus intranasally into ferrets. They use 2 doses. So, I keep getting this nonsense with dose. Every time I interact with people, they're always like, doesn't dose make it more severe? And no. And I'm going to show you these data. And, the other day I looked through all the animal data, and most of the time they don't use multiple doses, especially in non-human primates, because it's pretty expensive. But from what I see, you can give more doses. They get a higher fraction of animals will get infected, but they get the same disease. So here they give 1 × 10⁴, 10,000 or 100,000 PFU intranasally into ferrets. And the ferrets get infected. They can measure that by either doing nasal wash and doing plaque assays. Yay! They do plaque assays. And right after infection, in the group that get 100,000 PFU, the virus in the nasal wash peaks at about day 3. And then by day 7, it's gone. And by day 7, that's another song. By day 7, that's gone. By noon he was gone. That's the poem by John Updike, right? So you get this peak. And then if you give them 10,000 PFU, there's a little blip at 4 days, but they're basically not infected. So that's the threshold. 10 to the 4 is not enough to infect a ferret.
That's a pile of virus.
It's a lot of virus, right? Infectious virus.
10,000. 10 to the 5th is
a lot.
100,000.
And they're dripping it right in the nose of the little guys, right? It's not like it's moving around in the air on droplets. It's putting right in there, and it's not enough. Now, their susceptibility may be different from humans. But
one of the things that does happen with ferrets is that they can spread it between them.
They can. And
so
that's what they did.
It does require this huge inoculum to get the train rolling. Once one ferret has got it, you can put it into a cage with
another.
Uninfected carrots, uninfected ferrets, and
maybe they give them carrots.
Spread.
Yeah.
Well, right. I don't think we've seen it quite that promiscuous.
That would be impressive.
Yes, I would be amazed. But you could put it in a cage with uninfected ferrets, and they'll
transmit it. Now the thing is, this curve, 7 days. So then they say, what if we give this drug to the ferret? So they can give it to them orally. They have to put a tube in and put it in because they don't have pills. The ferrets won't probably take a pill.
I think from my experience trying to put pills in cats, I can imagine a tube would be very helpful. Yeah.
So they treat them 12 or 36 hours after they've infected them, and then twice a day for 3.5 days afterwards. Okay. So there's a lot. It's twice a day, 3.5 days. And that basically gets rid of the infection. There's a little bit of RNA that sticks around for a while, but no infectious virus. All right. And of course,
I liked that they looked at that because you do see the RNA sticking around.
It does, yes, but it's not infectious.
And you do a plaque assay, and there's nothing. The virus is not infectious, so the RNA sticks around, but there's no infectious virus.
And they look in other organs too, and they basically don't see anything. But that's the key. If you give this drug 12 to 36 hours after infection, it will do a great job. And I want to know, what about 48? What about 96? How far out can you go? You probably can't. I guess. You probably can't go far out. But if this is so with Tamiflu and Relenza, you need to take them 24 to 48 hours after you've been infected. And that's hard. A lot of people go longer out, and then the drug doesn't make much of a difference. They say, this is useless. I'm not taking this anymore. So I think 24 to 48 is tough. And but let's bear in mind that the whole course of infection in the ferrets is 7 days. It's not much shorter than people, right? It's 14 days in people. Yeah, it's 14. It's double that in people. That's significant. So if it's double 36 hours in people, that's 3 days, which means, you get infected, you start replicating the virus, and maybe you get tested because you think you had an exposure, or maybe you get tested because you just lost your sense of smell. You could hit within that window. Well, there's certainly scenarios where this would work, but I agree. But if I were doing this paper, I would have done the time course longer. It would be nice to see. I don't know why you wouldn't. Well, maybe they didn't have enough. But what you could do with this is, let's say you have an outbreak in a nursing home, you could give the drug to everyone. Right everybody. Before that everyone, and that could really help to stop outbreaks. So, but for community spread, if you go to your physician and you're not symptomatic, but you're just getting a routine test and you're positive, you could take this and it would be probably close enough to the time of infection. And you could prophylactically treat, not even necessarily in an outbreak, but people who are ICU workers and folks who are right up in the face of it at high risk. But that would depend on side effects and other things that you would need to look at in clinical trials. Yeah, how long, how often? You don't want to have to take it every day for months, right? So we don't know.
What you're saying is this is a party drug that you take the day after the party.
Right. Yes, it's a morning after drug for the party.
But as Alan said, you can study transmission in ferrets. You can infect ferrets, and then I think 12 hours later they put other uninfected ferrets in the same cage, and they all get infected within a couple of days, basically. And if you treat the initially infected ferrets with your drug, there's no transmission. The other ferrets don't get infected. So the implication is that, if you knock down the virus to nothing by the drug, you can't transmit, right?
There's no virus to transmit.
No virus to transmit. So that is quite interesting, but it does hinge on you getting the drug early enough to do that. Because if you're way past it, and it's not going to work. But I certainly think this is a justification to do trials in humans. Now, this has been through phase 1 in people already, and it seems to be safe. There are no side effects. And the drug concentrations in blood, that's one of the things they would measure in a phase 1 of a drug like this, which they don't do with a vaccine, is to measure the drug concentrations. They're high enough to be able to inhibit the virus, in cell culture at least. So it's promising. It is. Dixon, you seem to be making noises, right?
Well, I just wanted to add that if this paper had come out 6 months earlier, do you think the mink farmers would have jumped on it?
Yeah, you could treat your mink if you could afford it. I don't know how much it's going to cost. But Dixon, here's a better question. What if we had this drug in December 2019?
Oh,
huh?
Sure. Oh, sure.
Why didn't we? Why didn't we? Because nobody would support the bloody research. All right, this could have stopped the pandemic, in my view. That is Vincent's view. If we had this drug in December approved for humans.
Right. Not everybody on this show endorses that.
No,
view,
you don't have to endorse it. It's fine.
Yours.
But if this was developed for influenza, I don't know how, it didn't get very far, obviously, but it could have been developed for coronaviruses and it would have been ready. Now, I think this definitely has to go into a phase 2 in people, because even if the vaccines take care of the issue, there's still going to be outbreaks. You could treat them with this, and this will be ready for the next one. There's going to be a next one. And
the issue we just talked about with the vaccines being protective rather than sterilizing. You're going to need stuff like this sitting around for those people who didn't get the vaccine, or the 5% or so for whom the vaccine's not effective enough, or whatever. And we'll need this kind of thing for a while.
So, by the way, we didn't mention how this works. So this is a nucleoside analog, and you may think, oh, it must be a chain terminator, but it's not actually. What it does is templates as either a C, which is the normal base, or a U. So if it's a C, the next strand is going to be a G, right, on the other side. But if this thing is recognized as a U, then you're going to put an A in. So you end up having G to A mutations, and they say it just mutates the infectivity out of the virus, introduce too many mutations. It's a mutagen. So for that reason, they say others have done studies, and it's very hard to get resistance to this because you have so many changes in the genome. So that's good also, right?
What's the pharmacokinetics? How long does it last after you give it?
You have to take it twice a day.
Yeah,
I see me in the morning.
I think
you're a ferret.
Right. So the pharmacokinetics in this paper were based on, I guess, previous ferret studies, but it has been into humans in phase 1. So that's
a good question, Dixon. How long, does it last at a therapeutic level? Right. We don't know.
Exactly.
That's what you figure out.
Along the same lines as Vincent's comments about having a stuff around, it apparently is efficacious against a wide variety of RNA viruses, okay? Because, this is a pretty general mechanism. And so we're not just talking about coronaviruses. There could be spillovers of other RNA viruses where this could be a frontline candidate for managing a pandemic.
Let's not forget flu.
Flu too, yeah.
Right.
Sure.
I mean, that's my expectation for the next big pandemic was that it was going to be a bad flu, but that's still out there.
Yeah, I don't know why we didn't have this before. This drug has been around for a while. You can find it in the literature.
I know why. Money.
Yeah, of course. But Alan, aren't we spending way more money on the pandemic now?
We are. It's almost like preventing the problem would have been a lot cheaper.
Of course, we can say that with 20/20 vision in hindsight. However,
everybody in infectious disease has been saying this in prospect for the past 100 years, right? I mean, here's what you do to prevent the next pandemic. I've sat through entire conferences on this subject.
Right. And it's important to think about this because this is not going to be the last pandemic. And so it's not just about 2020 in hindsight. It's also how are we going to do things differently to change things for the next pandemic.
Yeah. I think Mark Dennison has worked on this, so has Ralph Barrick, and they have worked on it for a while. And Mark has always complained he could not get funding to do this. He said, I can show you all the grants I wrote about this kind of thing and couldn't get funded. So it's really unfortunate.
Because what were the odds of a bad pandemic coming along, right?
Pretty good.
Yeah.
Well, the corona's every 10 years, apparently, right?
Yeah.
So we have to keep TWiV going for another 10 years, see what happens.
See what the next virus is.
So this is 2030, that decade, the 2030s, right? Everybody, are you guys all in for this? You want
yeah.
I'm there.
As a matter of fact, I'm looking forward. This is sort of previewing my pick, but I'm looking forward to a TWiV from the moon. Okay. So that's going to take 10 years.
Yeah.
Okay. All right. Time to do some email. I have to take this first one because this was, we talked about papain
today,
right?
And things only New Yorkers will understand.
Said papain protease in terms of the viral genome last time, or very recently, and I got on this little spiel about how there used to be this juice bar on 86th and 3rd when I was a grad student. I used to pass it, and I go, "Wow, papaya and pineapple juice, right?" And both of them have proteases in them that help your digestion. In theory, I guess that's what it is. So Andy, who's an MD at the FDA, he's involved in cardiovascular devices, a loyal podcast listener. I love it. 86th Street and 3rd Avenue, where peptidase meets thirst. And he sends a picture of Papaya King. I forgot the name. And there's a link to a blog post, an underappreciated place in New York, the spot where allegedly the papaya drink was born, the corner of 86th and 3rd. Signs touted papaya's superlative health benefits, nature's own revitalizer, vitamin-packed health giving. And Andrew writes, alas, randomized trials are lacking. Thank you. It's great. That's it. That's exactly how I remembered it.
Yeah.
And if you go...
And there's a New York hot dog chain, famous New York hot dog chain called Gray's Papaya.
Correct.
Which was started by one of the people who founded Papaya King and left that to start Gray's Papaya, which serves a competing papaya drink. So, as well as hot dogs.
They have pictures on this website, and it's a really narrow, it's basically the counter and then 6 feet or so of space. Yeah. Everybody's crowded in there. And of course,
it's basically a food truck that's been installed in the wall.
I never went in.
It's still there, though. It's still there.
Yeah.
I don't know. You guys may not have ever ended up on the east side.
A couple of times.
But
it was at Rockefeller for 3 years. Come on.
And that's what 86th, this is actually, what is it? Germantown. Wonderful
Gray's
German restaurant.
Is I think it's on Broadway.
So you could go eat.
Gray's is on, they're in several locations, including West Side, but this Papaya King is on the east side.
But, you could go to, there were some great German Bavarian restaurants in this area. You could go and eat way too much heavy meats, and then you could go get a papaya drink and digest it.
And there's Kinsale's is right in that area, isn't it?
Kinsale's.
The bar that's got.
I think so. Yeah, I think so.
They've got a phone on the wall that's actually an internal line for is it Mount Sinai or Cornell that they're closer to?
Mount Sinai. They would be near Mount Sinai.
They're at Mount Sinai. Yeah. So they've got they're like the hangout for people who work at Mount Sinai, to the point that they wired an internal phone there. So if people, doctors got paged, they could pick up the line.
Yeah, there it is. It's 3rd Avenue and 93rd, 94th.
93rd.
Which is where I used to live, actually. I lived on 94th between 3rd and Lex. But it's closed. Yes, I remember Kinsale. It was right around the corner from us. Yeah. Yeah, so this brought back memories. That's why I'm kind of dwelling on it. Nostalgia, yeah. I think nostalgia is good from time to time.
As opposed to myalgia, which is not good.
No. And arthralgia, also not good.
That's also very bad.
Alan, can you take that next one?
Sure. Neil writes, we've seen an enormous number of infections at pork processing plants, slaughterhouses. And while we have seen cats, mink, and even tigers test positive for SARS-CoV-2, aren't pigs' ACE2 receptors nearly identical to humans? And as Vincent aptly points out with a reference, receptors are not the only determinants of infection. I would also add that receptors and infection of the livestock are not the only determinants of spread in a slaughterhouse, or even the primary determinants. The the reason we see this in meatpacking plants is that this is a work environment that has people shoulder to shoulder in a cold, wet space for long periods of time. And these are people who generally do not have access to much healthcare or testing or really anything else, because in many cases they're undocumented workers or other folks at the bottom of the economy. That's why we've seen outbreaks from slaughterhouses and meat processing plants, and it's not about the pigs.
The paper I linked to shows that pigs are not very susceptible to infection. So it doesn't matter that the ACE2 is nearly identical. Because once the virus gets in, there are other determinants, and pigs are not there.
Yeah, no,
Good
the
thing.
problem is the people.
Good thing.
The problem, and not necessarily the people who are catching it. The problem is the social structure that's put them in that situation.
Brianne, can you take the next one?
Sure. David, who's an MD, writes, to my new friends at TWiV, you have helped me live and experience a smart life every week during this pandemic. So lucky am I, and lots of others, to hear such intelligent discussion. Has this medium made all of us somehow closer? I do think that this is true. I am just a cancer doc, who rarely thought about virus until the last months. I still don't quite understand the endemic EBV, HPV, and VZV, and probably many others. I knew that I needed to learn more about virus, and then I found TWiV. Lots of comfort in hearing thoughtfulness from all of you. Very nice to be learning lots again. So smart and inquisitive you are. Vincent is the leader. He is a new day Socrates, and with his questions, I hear his kind style. He brings out the best in all. Brianne, the youngest of the group, a smile so sweet, and with deep interest, insight, and spirit. Drew.edu is a rhyme in itself. Rich, no need to smile, never dour or sour, lots of experience and perception of what really matters. He understands that there is a difference between what we can do versus what we should do. Kathy is sometimes quiet, but when she speaks, her ideas are profound. Each moment that I listen, I wait for her insight. So here I am in Central Massachusetts, new virtual friends and colleagues. I am very thankful for your TWiV project. We will survive. I'm really sad that I didn't get to say nice things about Alan or Dixon.
So, Dixon, do you want to read your addendum and then I'll read mine?
I'll read mine. I said, I guess I'm chopped liver. That thought makes me shiver.
Good.
And I would add, I'm also left out. How crass. Central once again forgets Western Mass.
It's good. It's very good. They belong with the poem.
Yes.
Good. Yes.
That's pretty good. Rich, can you take the next one?
Dora writes, I signed up some time ago for the Novavax vaccine trial. Novavax. Vaccine trial. Today they called and screened me. The call center agent had no answers to a single one of my questions and said they'd be answered at my first visit, for which I'd allegedly be paid $150, even if the answers to my questions led me to immediately walk out the door. I didn't even know whether to believe it. It turned out they couldn't have answered my questions. One question involved where I'd be tested for COVID if I became sick and where I'd be expected to get treatment. I never got any answer at all to this, not even we don't have the answer. This is an obvious confounding factor. If they need to diagnose it, then I may need to ride my bike 8 miles in the winter with a high fever or trouble breathing. If they go by my doctor's diagnosis, my doctor couldn't diagnose a boil on his butt. If it happens to test positive by the time the doctor's office and the lab mess it up, cowabunga. Now I ask, what if you don't know if you've already had the coronavirus, since I don't, and it only matters if you've ever had a positive test? I think that I and most people I know had COVID in March. Second, and most important, the trial is for 5 years. A vaccine should be available to high-risk people like myself in 2 to 3 months. I asked if they'd give everyone a shot at that point, or all high-risk people a shot at that point, or even tell us if we actually got the vaccine at that point. I pointed out that not only would I go get the vaccine if I hadn't already had it or couldn't determine if I already had it, but so would many people. I was told that we are free to leave the study whenever we want. After 2 or 3 months, they've not learned anything yet. I also pointed out in my email to them that less scrupulous people will get the vaccine elsewhere and not tell them. They'd gain the most valid information by vaccinating all of us at that point and then following us. If a third of people who were vaccinated subsequently get COVID, they'll know it works about as well as a flu vaccine. I was told they have no idea. In my mind, this must mean this study is not being well or ethically run. For certain, any results after the date when people get the vaccine, if they want to, won't mean a whole lot. Yours, Dora. So it sounds like Dora did not have a very happy experience with the vaccine trial. I don't fault at all the call center people for not answering questions. They're just taking calls. Sounds like she could have gotten maybe some better answers from the people running the trial. However, I mean, here we even stumbled, and apparently in the FDA conversation about the vaccines, there was a lot of conversation about what do you do with people who are in the trial, and it's a complicated question.
Her question about how she's going to get tested if she has symptoms. But I think we saw in the Lancet paper we did that you have a self-test kit. They'll send you swab yourself, you mail it in. So you don't have to ride your bike that 8 miles in the cold. So at least there's that, Dora.
Yeah, I think this is a mix of questions that that had easy answers, but she didn't get referred to the right person.
Yeah.
Because I'm sure, I know Novavax is setting this up, according to standards. And I'm sure the answers to this are available. And I think her email just got sent to somebody low level who blew her off. But that last question about what do you do when the vaccine comes out, the speculative question, as Rich said, that's something where there aren't good answers.
Although it depends, like as we heard from Brianne, they discussed yesterday that they would offer the vaccine when your time comes up to be vaccinated.
Yes.
And that's
I'm sorry. Yeah, there are good answers. They're not necessarily
great answers,
obvious or satisfying or definitive, and they are subject to some debate.
That's right.
Yeah.
Well, I'm not surprised. With so many trials going on, things like this are going to happen.
Sure.
Sorry about that. Dixon, can you take the next one?
Anthony writes, hello, TWiV. Shouldn't Dr. Fauci be advocating for a recovery-style randomized study to assess head-to-head the efficacy of Pfizer and Moderna vaccines and any others? If not, how come? Wouldn't such a randomized study be advantageous, especially for medical frontline workers and in persons and in nursing homes at this point in time? And then Vincent adds, see, mix and match Pfizer and Oxford jabs, the London Times, and it's got a big story about that. So are any of those comments valid, Vincent?
Well, somebody's in trouble. I can hear the sirens. Is that you, Dixon?
I can hear the sirens in your background too, Vincent.
No. This is Santa Claus. Are you guys kidding? This is Santa Claus.
It's Dixon. It's Fort Lee. I'm too high up here. So this Times story is they're going to put both Oxford and Pfizer vaccines. They're going to do mixing and matching to see if combining them makes a stronger immune response. They're not going to pit them against each other, but I think that's an interesting approach. And the other. Didn't you say that the Sanofi one said they would try them?
Sanofi is now pitching the idea of putting theirs in a comparative trial, which is a gutsy move, right?
Yeah,
I mean, if they end up having, so they're pretty confident in their vaccine, which they should be. It's using an established tech. So I think we are going to see these types of trials. The priority was on getting through initial trials with with the simplest design possible, because you don't want to compound it. I mean, imagine if we didn't have a phase 3 answer for either Pfizer or Moderna, we had a head-to-head trial between the two, and we had to deconvolute all of that before. No, you do these trials first individually. And now, as more vaccines come online, I think there's going to be more and more pressure to do these comparative trials. Are you as good as what we've already got? So yeah, that is definitely going to be in the works.
I don't understand the reference in this to a recovery style randomized study. Can you enlighten me? Is that a particular type of study?
Yeah, all these clinical trials have cute acronyms. So
the recovery trial was a specific trial.
I see.
Designed for COVID-19 to test many different things at once, right? And it was very flexible. You could add and take away treatments. Like it looked at dexamethasone, tocilizumab, convalescent plasma, monoclonal antibodies. So very flexible, very clever and flexible trial design where you could add and take away things along the way. So and you can compare things. Yeah. So
which
actually
is appropriate
is a website.
That's appropriate for a trial where you've got a whole bunch of already approved drugs.
These were approved drugs that they wanted to check for COVID. It's recoverytrial.net. You could see more about that there. I think the other consideration, Alan, is that we wanted to get something out there fast for frontline people and old people, and then later do the other kinds of tests, right? You don't want to be doing that initially.
Yeah, so you want to sign up 30,000 people for a trial, and you want to ask one question, and then get that answer, and then later on down the line you can sort out which is the best vaccine.
Right, and then you want to have something out there, and you can decide which of the things that are out there are the best later. But right now, we need to help some people.
I mean, in retrospect, it seems brilliant now, right? They did this mRNA vaccine, and it worked. They could do it quickly. And we were very skeptical at the beginning. We didn't know, right? And this focus on spike turned out to be okay.
They didn't know either.
No, they had to take a shot.
To be honest, and it's easy to confuse luck with brains. I mean, a lot of very smart people worked on this, but let's not downplay the role of a little bit of luck in there. We didn't know going into this that this was going to. But be the right answer.
A lot of technology that had been developed before.
Yes.
By the way, this idea of comparing vaccines reminds me of the polio vaccines. There were 2 oral poliovirus vaccines developed, one by Albert Sabin and one by Hilary Kaprowski in the 50s. And so they said, okay, which one are we going to use? So they did a match off of the 2. They did a trial where they compared the 2. This was run by Joe Melnick at Baylor. And the Sabin vaccine is slightly ahead of the Kaprowski. So those are the ones that we're using. And Kaprowski was always very upset about that. He said, mine should have been used.
In retrospect, has there been any comparison? Was Kaprowski's virus kept around? Because since that time, we've learned that there are issues with the oral vaccine, in particular shedding revertance to neurovirulence. And I wonder if Kaprowski's vaccine did the same thing. Maybe in the long run, it would have been a better deal.
Yes, it has been looked at, and I don't remember the results, but that has been examined. And I don't think the same attenuating mutations are present in the Kaprowski vaccine. So I can check. I will look. That's an interesting point. Now, of course, the Kaprowski vaccines were trialed in Africa, and he was blamed for, introducing HIV.
That silly book, yes.
It was silly, which is real wrong, but they said he grew up his virus in chimpanzee kidney cells, which he got from Africa, which had, SIV. It's just a crazy theory, but it's not true. It's not true.
So, Vincent, how many different kinds of flu vaccines are there? And have they ever put them head-to-head to see which ones are more efficacious or not? Because I just got my flu vaccine, and I asked the guy giving it to me which one I was getting. He says, "Damned if I know."
Damned if I know.
Oh my gosh, that's bad care. You should always be able to get that. It's on the vial that they're pulling it out of. I mean, they should certainly know, and they should have. I think there's usually a sticker or something that they on the form that you sign. It tells you.
Yeah.
It should tell you which one to get. Anyway, in answer to your question, yes, in fact, there have been a number of comparison trials because you can do this in any flu year. because people get, most people in America are going to get the Sanofi vaccine, but a lot of people are going to get, nowadays, there's the high dose for folks Dixon's age. There's the live attenuated. So people do get lots of different vaccines. And there have been a number of studies done where they look at comparative efficacy, and there are figures on those that show you can point to a study and say, well, flu mist has like 80% efficacy versus, flu block, which had 60% efficacy that year. And that's the critical thing with flu vaccine, because then the next year you find out that the flu mist had much, much lower efficacy than the injected vaccines. And so things change around and recommendations change. And the thing is that because you've got different strains of flu every year, because you have different conditions every year, the vaccine's being adapted for each year's flu strains, it's ever changing. So yes, the studies have been done, but the answers are not informative for next year necessarily.
I just remember I actually have the Kaprowski vaccines in my freezer, at least some of them, because when this controversy arose, they said he grew up the virus in chimpanzees from Africa, and that introduced HIV into the human, or SIV into humans, and then it became HIV. So they found some of the vials at the Wistar Institute, which is where he had done this. And I don't know, somehow I got involved in the committee that was looking at this, and they said, okay, Racaniello, you're going to aliquot them and code them, and then we're going to send them out for PCR to see if there's any HIV, SIV sequences in them. That'll answer the question. So I remember, well, the answer is, don't you want me to tell the story first, or you want the answer?
Of course.
So I met the director of the Wistar Institute on exit something on the New Jersey Turnpike in a Dunkin' Donuts or something in the parking lot. And he gives me this styrofoam box, right, with dry ice. It's like we're doing a drug deal. I put it in my car. I drove it back here, and I aliquoted it and coded it. And then I met him again another day and gave it back to him. And then he sent it out to a variety of labs who looked, and including Svante Pabo. He did the mitochondrial DNA sequencing as well as the others to see if it indeed had been grown up. And anyway, the bottom line is it had no HIV or SIV in it. End of story. And they published this paper, and that was the end of it. But I wasn't on the paper. I wasn't even thanked in the acknowledgements for coding.
They didn't even pay you in $20 bills, unmarked in a paper bag. It was
That's the way things are usually done at an exit on the Jersey Turnpike.
Well, we saw the Sopranos. That's.
But isn't that funny? I met the guy on the New Jersey Turnpike.
Meet at the Vincent Lombardi rest stop.
Exactly right.
I don't know why we didn't send it. I'm not sure, but anyway. Okay, let's do one more round here. Brendan writes, after listening to the last few episodes and mentions of vaccine distribution, I found myself wondering whether we are thinking correctly about how we classify most vulnerable. As has been emphasized in recent episodes, especially when referring to the long-term COVID and morbidity associated with clotting, should we be thinking of the demographic of potential patients and with valuable life years first, in addition to frontline workers? This idea of vaccinating immobile and less mobile elderly individuals may save lives, but as you have discussed, life doesn't exist in a dichotomy of recovered and deceased. The morbidity of those in between can be the most significant aspect to this pandemic. I was wondering if you could address whether vaccinating the general population first, along with healthcare workers, makes more sense as a top priority, since they have the potential of going from household to household, group to group, keeping viral prevalence high and prolonging the pandemic more than an elderly person in a nursing home. It seems to me that the ambulatory person walking and traveling around town has the potential of a much higher R0 than most. I don't mean to minimize protecting the elderly, but the people who still support the economy, provide childcare and care for their families, and make up the bulk of spreading infection may be the most important in getting us out of this mess and keeping the long-term consequences minimized. Thanks for this great podcast. This is my favorite connection to the medical world, since I'm a former MD and miss great knowledgeable discussions. And Brandon is at Finhorn Illustrations, LLC. So my thought is, the vaccine is probably not going to prevent transmission. So if we adopt your proposal, to immunize the mobile people, it's not going to may not stop them transmitting it, right? So I would think then, why don't we just save lives? And that's why the elder. That's my feeling. I don't know. What do others think here?
I hadn't thought about that particular aspect, Vincent, and I think that that's a critical point. Beyond that, I'm not the statistician or epidemiologist. The goal here is to save lives, right?
Mortality,
and that is beyond my ability to calculate what these. I mean, he raises an interesting point.
Yeah, it is.
But I can't really parse the numbers.
Right. Well, I think that I would agree completely that because the vaccine is largely protecting against disease, we should be thinking about who needs to be protected from disease. And that does seem to be elderly individuals. If you look at the plans that put me way down far in that line, you do notice that after elderly individuals and healthcare workers, there are other groups that are prioritized, like those who are working in essential jobs, who are going to be in contact with members of the public and things like that. So I think some of this idea of thinking about other groups is there as part of the tiers of those getting vaccinated. But right now we need to protect those who are going to get severe disease, especially since maybe we could have done a little bit of better job protecting them in the first place. It's sort of like we owe them some protection now.
And also, again, I'm not a statistician either, but I'm pretty sure that if you run the numbers on this, you find that even if you express it in something like disability adjusted life years, which I think is what Brandon is getting at, that's kind of the standard currency in these public health discussions, you would still come out in favor of vaccinating the elderly very early in the process, because the number of disability adjusted life years you lose for somebody who's 85 may not be as many as you would lose for somebody who's 25, but the likelihood of losing all of those years for the 85-year-old is drastically higher. So the population that's going to be affected, the elderly population is going to be affected, the high-risk folks with other medical conditions, heart disease, whatever, those folks are going to be, in really bad shape at a very high rate. And the young folks getting sick is a much, much rarer event. And so I think even if you did it on a basis of DALYs, you'd still come out in favor of this. And as others have pointed out, there's the issue that the vaccine is probably not going to provide sterilizing immunity across the board. And the issue that the population, the working age population, is immense, right? So if you say, okay, we're going to put people who are between 18 and 50 at the front of the line. Well, great. That's how, the majority of Americans probably. It's a huge, huge number of people. So by the time you get through that, the people who are at highest risk of dying have probably already done so. Whereas if you narrow it down and you say we're going to start with these smaller populations, the elderly, the healthcare workers, yada, 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 then you can actually get through those groups before moving on to the general population.
Yeah, there's some other issues here, too, that need to be addressed, and that is people of color, Native American populations, sure. Those underrepresented, highly marginalized people are really getting the shaft in this infection. And I would put them very high on the list, and I don't see any indication of that whatsoever on anybody's list. But, I mean, I would gladly step aside to see someone from the Navajo Nation take my place, for instance, because I know that they're highly susceptible to this infection, and they have a lot of behavioral problems with regards to when someone dies, for instance, the entire group shows up to celebrate the death and the life of that person, I should say. And in doing so, they all now are more susceptible to catching the infection. I think there are so few of them. Why can't they be number one on the list? Just do them first, along with the healthcare workers, of course. But, I saw several PBS programs on this, and I'm thoroughly convinced that people of color and Native American populations are very, very underrepresented in terms of opinion as to what we should do. And so that's my 2 cents worth.
Sure. There is, this is a different angle on that, but there is one marginalized group that has been discussed as being high priority, and that's prison populations.
Yeah, that's right. That's right.
Which makes a lot of sense because it's a very confined place, and we've already had prison outbreaks that have had, in some cases, very high mortality rates. So, yeah, I think that's a group that probably ought to be at the front.
I believe you are next, Alan.
Wait a minute.
Okay.
Just before we leave
Sure.
this, I want to point out that Brandon's illustrations are quite something.
Oh, yeah?
I linked to his site there. A lot of fish.
Fish, Dixon,
I'm gonna
fish.
go look. I'm gonna go look. I will go look.
Not now, Dixon.
Oh, these are neat.
You're working.
These are cool.
I'm a working stiff.
They even have a fishing pole, Dixon.
Listen, that's not unusual. A lot of people fish.
And it looks like there's a coloring book.
Many people fish, but very few catch.
I like the crab with what looks like a screw pile style lighthouse. We had those on the Chesapeake. Anyway, cool stuff. So Junji writes, and I thought in computer science we have a lot of acronyms. Follicular dendritic cells adhere to fibronectin and laminin fibers via their respective receptors, and links to a paper. And I haven't read this paper, but if you're in the immunology literature, then yeah, they are world-class acronym generators, and I'm going to let Brianne take it from there.
Oh yeah.
Y'all are up there with the military. I mean,
immunologists are known for jargon in general and different acronyms in particular. And in fact, even in what exactly you're doing in immunology, sometimes the acronym changes what it stands for. APC can be an article processing charge, an antigen presenting cell, or the flow cytometry dye allophyocyanin. And so you almost have to really know the context of that acronym.
Nice.
Because we have so many.
If you just look at this paper, you don't even have to read it. Okay. The acronyms just jump out at you like a blizzard.
Yes.
Yeah. I think immunology, as Brianne said, is pretty guilty of that.
Yeah, it is.
Brianne, can you take the next one?
Sure. Wayne writes, Japan enacts law to make coronavirus vaccines free to residents, from Japan Today. And he gives a link. He says, Dr. Vincent, hope all is well. TWiV is truly a bright light in this morose world. Vaccinations in Japan will be free to all.
Nice. I think that's great,
that's
right?
great. Yeah.
Who's paying for them in the U.S.? Our health insurance?
The vaccinations themselves are... My understanding is the vaccinations themselves are being paid for by the federal government. It's not clear where the funds are going to come from for some of the distribution.
Oh, that's right.
Okay, which falls on the states. Well, yeah, sure. I mean, in Japan, I'm sure it comes out of your pocket as well, ultimately. Okay.
That's right. I read somewhere an article, the vaccine development has been paid for by the federal government, but all distribution...
Except Pfizer.
All distribution is up to the states, which is like 8.4 billion, and many states can't handle that, as you know. So They would like some help. That's a big problem. Rich, can you take the next one? That's Tony.
Tony writes, Dear Truthers, in TWiV 687 and other podcasts, you have said you are preaching to the choir. Please don't take this as a criticism, as I love the show, but the reason you have this problem is because you are mere scientists and not social media influencers. In order to get the truth out, I have a plan. You will hate it, but it should work. And that's all that counts right now. I think you should have another show called TWiM, This Week in Moronology. Invite conspiracy theorists and anti-vaxxers as guests. Then you can rip them to bits with science fact and not science fiction. This would attract far more listeners and subscribers, and you could easily go viral. Many people would then go on and listen to your regular stuff. Yes, it would be a gimmick to some extent, and not so cozy for you guys, but this is a war that must be won to save lives. I picked the name TWiV as a joke, and you could choose something less confrontational. Thanks, Tony. That's not an activity that I would enjoy.
No,
and, I'm not practiced at the kind of slime that the anti-vaxxers and conspiracy theorists do. I went to a vaccine advocacy thing at the Capitol in Austin some time ago, and I was confronted by a reporter, these are air quotes, from InfoWars. And he blindsided me because I didn't know who this was or what it was. And it was extraordinarily uncomfortable. I chose to talk to him for a little while, and it's impossible. Okay. It's like having a debate against certain politicians in our experience. Okay. First of all, you can't get a word in edgewise. Second, the truth doesn't matter. There's no reasoning. Okay, so forget it. It's all it does is give them airtime.
And I would just say, Tony, I really like all of the tech I have here. And I think that if I was involved in any of that, it would get broken quickly. So I think that might be a bad idea.
You'd be punching it, right?
Yeah.
So the real issue going around the country asking to be guests on far right information shows, in quotes information, that was quotes too. And he's willing to put himself up against those people, and we both, all of us, gave him our deepest sympathies.
Who is this you're talking? Peter Hotez.
Peter Hotez. Yeah. Right. The issue is Peter Hotez is putting himself on places like Fox News, which is far right, and they do a lot of disinformation, but they are kind of mainstreamish in the sense that they do have millions and millions of listeners. And in many parts of the country, they are the primary news source. Whereas something like InfoWars is so far out there that you're reaching the people who have already drilled down through Google's algorithm, way down into the rabbit hole that maybe they initially got sent on from Fox News, but now the blinders are fully on. And the problem with having folks at those extremes on a show is that you legitimize them. Which, of course, Fox News does. They have the InfoWars people come on and provide commentary. But just talking to them raises their profile and their argumentation to a level it does not deserve. And the reason I mentioned the story of the tar baby is that the more you argue in public with these people, the worse it's going to go for truth, because they're not interested in truth. They're not confined by facts. They're not limited by knowledge. They're not restricted by any of this. So they can just maneuver in unlimited space and make crap up, and you'll never win.
All of that having been said, Tony, I appreciate the sentiment.
Thank you, Tony. Yes.
Yes.
I think I agree with you, Alan, that it legitimizes them, but I do think, and maybe what you're saying is you can't counter them because they will just twist out of any logic that you use.
No, you can go to some place like Fox News where they have to maintain some semblance of reality in order to, keep their advertisers. But once you get down further into these extremist pockets, just forget about it.
All right. Okay. All right, Dixon, take this last one.
Ken writes, Dear TWiV, the temperature in Chicago is now 28F minus 2C. My son attends the University of Illinois at Urbana-Champaign, the same university which implemented a twice-a-week mandatory saliva test for all students, faculty, and staff who were on campus. U of I has a cumulative test data website. Here she gives the link. He gives the link, which allows you to view and download information such as number of cases, case positivity rate, and number of tests run. At this point, there have been 4,195 cases discovered from this testing, which has started from July 6th, with the daily number of tests ramping up around August 17th. My main question is if any of you are aware of any data collection which utilizes these test results to see if there's any reflection within any of the 4,195 cases, or I'm sorry, reinfection within any of those cases. And I meant to say that. This type of mandatory testing provides a great opportunity to see if there are any reinfection, and if so, what the timing, severity, and frequency would be. Aside from the U of I, there should be data from the saliva direct test developed for the NBA bubble, COVID testing for the NHL bubble, and non-bubble Major League Baseball and the National Football League. Obviously, this data should be anonymized, but a great opportunity exists to shed some light on the questions related to reinfection. One additional request: the next time Kate Rubins comes on the show, please ask her to let us know the temperature outside the space station. Actually, we could give you that right now. It's 3 degrees above Kelvin.
I think it depends on where in her orbit she is.
Yes,
exactly
is she on the
right.
light side or the dark side?
Yeah, I meant to add that. You're actually right.
Probably next time we have her on, she'll be on the ground,
Right.
Could be.
I don't want to monopolize her time. Other people probably want to talk to her.
Oh, I'd like to monopolize her time.
I mean, she is pretty interesting. It is pretty great to talk to.
Well, we could say towards the end of her stint, we could have her back to say, "What did you learn?" Maybe she'd like to do that. What did you say, Dixon? What did you just ask?
When she lands, we could have her on as a guest.
Well, we were planning to do that anyway. When she landed, or give her a few weeks to get her legs back.
Get used to gravity again, yeah.
But it would be also interesting to have her at the end of her 6 months and say, all right, what'd you learn? Someone sent in a picture of her and sent to TWiV a picture of her in front of this little chamber with plants in it.
Radishes. Those are the radishes.
Yeah, it was really cool. It was this little box. And there were just wires everywhere. And I responded to the guy, obviously, cable management is not a priority. There's just tons of wires everywhere. And then there's this laptop just kind of floating because it doesn't go anywhere.
Yes.
Wow. Crazy stuff. All right. Time for some picks of the week.
Before that, I will just say, I think that's a great question, Ken. I am not aware of any such thing.
Yes.
I'm not aware
of
any such things, but I think that's a great idea, and I would love to know about it.
Yeah, I agree. I'm not aware of any, and someone may be actually using it, but the data are cool. But it's anonymized, right? And only you know your results. So you'd have to design a trial to. Well, if it's a clinical trial, you could get consent, so it's not anonymized anymore. So you could
Vincent,
do that.
has there been any improvement in the time frame between the time you give a saliva sample and the time you get the results back? What's the shortest period of time that can elapse between you give the saliva, they bring it to the lab, they start the test? How much?
15 minutes. Here in New York City, MD, you can go in 15 minutes to have a lamp test. That'll do it.
Why the hell don't we do that?
Yeah, the Abbott ID Now, which came out months ago, does 15 minutes. It's a little less accurate, but it doesn't matter. We're doing it. You can go get it done at City MD, Dixon.
You think?
Yeah, in the morning when I drive in, I pass one on Route 22 or Route 22. Someone said to me, why do you say respiratory instead of the correct respiratory? I'm like, what is it correct? According to whom? I was trained respiratory by physicians and scientists. Sorry.
Yeah.
And it's a City MD. There's a line going around every morning of people waiting because they want to get their 15-minute test so they can go to work. Yeah, it's being done, Dixon.
We should do more of it.
Okay, you can. Go ahead.
No, I mean,
I don't know what your point is.
My point is that I'm serving on a local committee here in this building. There are 300 units here. There are X number of people here. Why the hell don't we have that testing on foyer?
Let me tell you what to do. You use the Homeowners Association dues to buy a unit. It's not that expensive. You set it up and you run the test yourself. You can coordinate the whole thing because you're a scientist. You can do it. Why don't you propose it?
I'm going to. I will.
You can do it. You can buy one of these kits. You can buy one of them.
Send me the information, and I'll be glad to do that.
You bet. Now, since I have your attention, Dixon, why don't you tell us your pick of the week?
I will. And it's actually the 2020 photos, a year like no other, the New York Times. And when you look at this, these are not pretty pictures of animals or bees or trees or flowers or anything else. These are pictures that tell you something happened at that moment. And, photojournalism is an incredibly powerful tool. And the New York Times is a highly selective newspaper that doesn't print all the photojournalist photos that they receive every day or every week or every year. And these are the highly selected years worth of photos that takes you step by step through what happened last year. And they're emotional, and they're hard to look at, some of them, and they tear you apart, and others put you back together again. So it's really a time capsule for the last year.
Yeah, there's some.
Remember, oh my gosh, that happened this year.
Yeah,
exactly. It's been such a long year, and we've been in isolation for such a long time. You feel like you were just let out of prison or something.
Yeah, some great pictures here. It's really nice.
Yeah, it's remarkable.
Cool. Brienne, what do you have for us?
So I have sort of something that is related to a past pick on science and cooking. So this is another of the science cookbooks that I have called Cooking for Geeks. This one is really fun. It contains experiments. It has a lot of graphs and data, and really explains the background of quite a few different cooking techniques. It's one that I think a lot of people who are interested in science would find really exciting and interesting and fun. It's just a fun one.
Cool.
Brienne, I'm looking through the introduction here, and they have a great Venn diagram that describes what a geek is.
Yes.
Okay? A geek is a mashup of curious and smart, as opposed to a dork, which is a mashup of curious and awkward. Or a dweeb, which is a mashup of awkward and smart. They don't have the intersection of all three, smart, awkward, and curious. I'd like to know what that is. At any rate, this looks great.
Yeah. Well, I just like the fact that it actually, is for the kind of person who wants to do an experiment on the difference between taste and smell and what happens if you treat your apples and potatoes in different ways. How can you suddenly change what you think it is?
Cool.
This looks cool.
Very good. Alan, what do you have for us?
Well, this is actually riffing on a pick that Brienne previously had, and I didn't realize she was going to be on an arc with it. So we are now on this arc together, Brienne.
Perfect.
I got this, I think I got it last year for Christmas, actually. This is to cooking what Fields Virology is to virology. So this book is called On Food and Cooking by Harold McGee. It's not the kind of thing that you read cover to cover unless you're, enrolled at the Culinary Institute and this is part of your 4-credit food science course, which it may very well be. But it is the kind of thing you pick up when you're in the kitchen and you're wondering, why did this cake collapse in the middle like that? What exactly makes a cake rise? I can look that up, and you can find out why it's different than bread rising, and the different chemical leaveners that have been used, and the difference between rising with a gluten-based bread dough and rising with a low protein cake flour type of batter. And you can drill down to that just as deeply as you like, or if you've got questions about different types of cheese, this thing will go as far as you want into the food science and the chemistry, and it's just a great reference to have around if you're curious and about this sort of thing.
If you're not a dweeb,
if you're a food geek, this is a great book to have on hand.
Cool.
It looks like the link on Amazon has got the whole book.
Oh, well, maybe you don't have to buy it.
It's a pretty big book.
Yeah,
it is like a very, very big book.
Like over 800 pages.
That may be the whole thing. Oh, page 80. A lot of pages are not included, it looks like.
I'm sure they don't have the whole thing there. Cool.
Yeah.
Food arc. Yeah, I could dig that.
Food science.
Rich, what do you have for us?
I'm still on my space arc.
Yeah.
Okay.
I can see.
My pick is the NASA Artemis program, which I was not aware of until recently, and I'll tell you why. I won't tell you why I wasn't aware of it. I don't know, but I can tell you why I became aware of it. So this is basically a latter-day Apollo project. We're going back to the moon. And the name of the project is Artemis. Artemis is a significant name because Artemis is the twin sister of Apollo. Artemis also has connections. She's the goddess of the moon. Artemis is also female. And as part of this project, we are going to land a female on the moon. And this particular site, plus I also linked a wiki site because I used both to try and figure out what this actually was. Together, these two resources tell you about a series of missions. They're planning about 6 at least missions that put people back on the moon in what they say a long-term and sustainable fashion. So we're going to have a moon base. And it involves actually the construction of a lunar orbit space station as well as an intermediate platform in the whole thing. And it uses a new bunch of hardware developed by NASA that includes the Space Launch System. That's a heavy-duty rocket and boosters that will launch all sorts of different configurations of crewed and cargo payloads. And a new crewed capsule called the Orion capsule. It's actually been in development for quite a while. And the first mission will be a robot mission that sends people is an unmanned mission that just goes to the moon and back to test the equipment. And then there's a manned mission that goes to the moon and back without landing to make sure that's okay. And then mission number 3, they will land people on the moon. And in the meantime, they're going to be constructing this lunar orbit space station to house people and equipment and stuff as an intermediate step. And ultimately, we'll have bases on the moon where people can stay for up to a month or maybe even longer. And the reason this came to my attention is that just a few days ago, the Artemis team of astronauts, 18 astronauts, was announced. And lo and behold, there on the list is Kate Rubins.
Hey, cool. I guess she's got the right crew.
I think Kate's going to the moon, dude.
Nice. Very cool. All right, but then maybe she can do a TWiV from the moon, right?
That's what I said. Yeah, I'm waiting.
So we're talking about
that would be awesome.
It's too bad her name isn't Alice.
Yeah, right.
Right.
But is there money for this, Rich? It's going to happen for sure?
Yeah.
Really?
Well, it's going to happen as much as any sort of NASA thing is going to happen, right? These plans come and go. There were precursors to this that came and went, but there's they change. The plans evolve. Okay, but yes, there is funding for this. As a matter of fact, if you look at the NASA site, part of it is the funding, and they tell what they need, how much it's going to cost, blah, 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 blah. And what is proposed so far, I believe there are funds committed for it.
Cool. Very good. Love it. All right. My pick is a very nice opinion piece in the New York Times by David Quammen. He's been on TWiV a couple of times and on TWiVo. And he writes beautifully. And, this morning, the name of this opinion was A Few Words on Behalf of Bats. But when you link to the article now, the title is Plenty Can Be Said for These Blood-Sucking Disease Carriers. Bats are likely the source of the virus causing a pandemic. They are also magnificent. So they changed the title between now and this morning. Isn't that interesting? Anyway, but it's a lovely article. He writes so beautifully.
It is very nice. I read this. Very nice.
It's all about, hey, it's not their fault. They didn't purposely do this. They're just beautiful critters. In the first paragraph, the order of animals known as Chiroptera, the bats, enjoys a mixed reputation among humans. I'm putting this politely. They have been calumniated and abused for centuries. Calumniated. What do you think of that word? Huh?
Got nice pictures of bats, too.
Yeah.
You make false statements about them.
It's a very Shakespearean term. But but yeah, it's David Quammen's beautiful writing, and it's a nice long explanation of why we should care about bats. And it's more than just they're beautiful animals; they're important animals and widespread.
Yep.
And yeah,
it's good writing. It brings in some of his travels and so forth. Very nice. Nice job. All right, that'll do it for TWiV 693. You can find the show notes at microbe.tv/TWiV. Send your questions and comments to twiv@microbe.tv. If you like what we do, you could support us financially. We depend on your help. Microbe.tv/contribute. Dixon Despommier is at Racaniello.org, thelivingriver.com. Thanks, Dixon.
Pleasure.
Pleasure as always.
Absolutely, as always.
Very good. Rich Condit's an emeritus professor at the University of Florida in Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Brianne Barker's at Drew University, BioProf Barker on the Twitter. Thanks, Brianne.
Thanks. It was great to be here.
And Alan Dove is at alandove.com. On Twitter, he's Alan Dove. Thank you, Alan.
thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and all of you, of course, who support TWiV. Thank you very much, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.